Good morning, everyone. Thank you so much for being here. I have really been looking forward to this. I've loved the research that I've done this summer and I've been looking forward to sharing this information with you all. So this summer I was able to work with a COP lab here at CU Boulder, and I was able to study or research basically how to culture and identify soil bacteria. So first I'd like to kind of zoom out and we'll take a look at the bigger picture. Okay, so just to put my research this summer into context. So the larger problem at hand is that we are facing a global crisis of antibiotic resistance, okay? And so the solution to this would be, well, let's find more antibiotics. And we would want to focus on the bacteria in soil because they naturally produce antibiotics. The caveat, though, is that soil bacteria is really hard to culture. They're really picky, and currently only 0.3% of soil bacteria have been cultured and identified in the lab. So this is where I come in, because my research this summer was about how to encourage more diverse bacterial growth. So specifically, I wanted to look at what would happen if I kind of gave the bacteria 4 different carbon sources, so 4 different types of food, basically. So I changed up an ingredient in the chemical cocktail that they grow on, and I wanted to see how that would impact growth and if it would increase the diversity of growth. From there, I would want to test if those bacteria were bioactive against E. coli. So basically, can it fight off another bacteria like E. coli? And then we would want to identify those bacteria by sequencing their DNA and also analyzing their lipid profile. Okay, so as for the methods, again, I mainly was worried about, or focused on rather, culturing and identifying. So for culturing, we made minimal media. Media is just a chemical cocktail, again, that we kind of use to grow the bacteria on. Minimal is just in regards to the nutrient content, so this isn't, other medias are very nutrient rich, and we kind of wanted to tone that down a little bit. We used a method called serial dilution, which is basically just, let's take this soil water, which has so many bacteria, and let's do a series of dilutions so that when we're growing colonies on a plate, that petri dish is not super overcrowded with bacteria. And so here's an example of what that looks like. You can see on the very far left, we have way more concentration of colony growth on the plate. And then as we move further to the right, very little colony growth because this concentration was far less. And then we also wanted to isolate the strains. So this just means that we take the dilution plates, all the colonies that pop up there, we want to streak them out, and then we want to, from there, create a separate patch. The reason we want them to be isolated and not like growing in a buddy system is because when we amplify and sequence their DNA or analyze their lipid profiles, we don't want mixed results. We only want to focus on one little guy in there. Okay, so out of all 4 of the carbon sources that we tested, sodium acetate was the one that the bacteria liked the most. The way I thought of this was fussy toddlers with chicken nuggets. Everyone's happy, it's a great night, it goes really smoothly. The one that had little growth, or actually 0 growth in this case, was the sodium bicarbonate. Brussels sprouts is what I'm thinking for this one. Toddlers and Brussels sprouts. It's just, we weren't having a great time. Nothing really happened there. There was no progress. Out of all the strains we were able to identify from here and test the bioactivity of, 0s, straight across the board. None of our strains happened to be bioactive against E. coli. And this was certainly disappointing. However, my mentor did have a frozen strain from her work last summer, which was identified and already proven to be bioactive against E. coli. So if you take a look at this patch plate here, there is, is there a laser on this? Oh, let's go. This little halo around this patch right here, this indicates that the bacteria in that patch is fighting off the E. coli that's been spread onto the plate. So this is cool, first of all, but B, it also meant that I was able to do some more testing, which was really exciting. So my time in the lab didn't have to just end because we didn't have more time to really grow more colonies or anything. So specifically, the further testing I wanted to do was to analyze the lipid profile. I'm gonna break this down into two parts. First, I will tell you kind of what a lipid profile is, and then I'll reiterate the purpose of the lipid profile, specifically in the context of my research. So lipid profile is basically, you have your bacterial sample, you go through a process of extracting the lipids from the membranes of the bacteria, okay? And then you use an instrument called a gas chromatographer, or a GC, and this instrument is basically able to take the sample and break down, like, here are the lipids in the membrane, and here are the specific fatty acids that make up the lipid. The way that I thought of this was, it's like a wine tasting. So you pour a vial of lipid sample for the GC. The GC wafts the lipid sample and says, am I detecting a hint of an 18 carbon long chain in this lipid? And then the GC takes a sip of the lipid sample and says, oh, is this a 16 carbon chain in the lipid? And this is pretty much the breakdown. So that was part one. Part two is, again, reiterating the purpose of a lipid profile, which is to identify or characterize the bacteria. So this is kind of like, our fingerprints are really unique to us, the way that a lipid profile is really unique to an organism. This strain, this bioactive strain that we had from last summer, was already identified by 16S or DNA sequencing. We knew the genus, which was Pseudomonas, but we didn't know the exact species. And so what I used the lipid profiling for was just to confirm that this was indeed a Pseudomonas strain. The way that I did that was I compared the lipid profile of our bioactive strain to a known lipid profile of another Pseudomonas strain. There were two separate samples. One was from an environmental culture and one was from an infection culture. So these leftmost three bars basically are the three Pseudomonas. And you can see in the levels of the 18 carbon long chains right here in the dark blue, there's strong similarities across the board here with the Pseudomonas, specifically between the one that I wanted to confirm whether it was Pseudomonas and then the infection sample. And then also these other two, 16 and, both are 16 carbon chains that are, those are basically the common fatty acids that the lipids share. I also wanted to compare this to another bacteria, K. vers, just to really contrast the difference, I guess. And so you can see here, this dark blue bar at the bottom of the K. vers, the 18 carbon chain, so low, so little. If you waft that vial of lipid sample, you're probably not going to detect the 18 carbon chain, right? So this is just to highlight the difference between the lipid profiles of two separate bacterial strains. This data basically confirms that yes, our strain that was bioactive from last summer is indeed Pseudomonas, and definitely K. vers. Moving forward, I would really like to test whether this bioactive strain is also bioactive against yeast. The reason is because yeast is eukaryotic cells, right? And we know that it can fight off prokaryotic cells, bacteria like E. coli. To loop this back around to the antibiotic resistance crisis, if we want to find a viable antibiotic, we want something that will fight off bacteria, prokaryotic cells, but will not fight off eukaryotic cells, because that's what we are made of. So we wouldn't want a medicine that would kill the bacterial infection, but then also harm us, because that would be useless. We also could go ahead with continuing to vary carbon sources, or as a further extension, maybe even varying other ingredients in the chemical cocktail or the media. So the sulfur or nitrogen or phosphorus sources. A broader extension of this research would be to explore the difference of how bacteria grows when we isolate it the way that we did, or how it grows in the symbiotic relationships that it actually naturally experiences in the soil. So with other bacteria or with byproducts that the plants around it are putting out. And that's that. I have had a wonderful time learning all of this. This was brand new to me, and I would really like to know what's coming to mind for all of you.